I know some oncologists, and one thing I can tell you for sure, and I get sort of emotional about this, those folks, if there were a cure for cancer, they would make sure that people got it.
I can't think of any more human activity than conducting science experiments.
The game I play is a very interesting one. It's imagination in a tight straitjacket.
The beauty of a living thing is not the atoms that go into it, but the way those atoms are put together.
What I always think should be the basis of education is not answers, but questions. We should teach kids how to question.
Well, guys, my name's Kieran Scott, and I work here at Western Sydney University at the Ingham Institute in Liverpool. And I'm a biochemist and molecular biologist, and who's wound up in cancer research.
Could you tell us a bit about your journey, about how you went from biochemistry into oncology, because that's an interesting transition.
Yeah, so I trained in biochemistry many years ago, and so a lot of the biochemistry I've learned, there are two things that happen. A lot of the biochemistry is really fundamental to what we do today, but a lot of it also has been improved on enormously. So I trained in general biochemistry in the days when protein sequencing was the thing. And so that's quite a long time ago. And guys like Sanger got a Nobel Prize for inventing how to do that, and then he went on to get a Nobel Prize for inventing how to sequence DNA. So that's a long time ago.
Yeah.
Because one of the things that I really scares me when I go into the lab today is when I did my PhD in genetics at the ANU. I was working in plant biology then, plant-bacterial interactions. And I got my PhD for sequencing 3,000 base pairs of DNA from a bacterium. It's the sad thing for me is every day when I go into the lab now, there's this tiny machine, and it's only about this big, and it sequences entire bacterial genomes in less than 24 hours.
Yeah.
It's interesting. We were at the Garvan Institute a few days ago, and they have literally a sequencer that's probably that big that you could potentially even connect to your iPhone.
These things are made by Oxford Nanopore Technologies. I don't know if you've heard of them.
That's right. You see, those are the things I read about. But
They've got a mini ion now, which basically it's like a sequencer that plugs in the end of your iPhone, and you can sequence out in the field.
And you can sequence strands that are longer than 1 megabase, so 1 million base pairs in one read,
Well, picture this, guys. I'm, very long time ago doing my PhD in my white coat, carrying around a sequencing gel that is 1 meter long, and so I then hook that up to an electrophoresis machine, and I load my samples, and then 2 or 3 hours later, after the magic has been done.
You have to do your little dance, don't you, as well?
Right.
The rain dance.
After the magic has been done, I get to take the gel and expose it to X-ray film, So that either says I'm very old or technology has come a very long way. I prefer to think it's this.
The latter, for sure.
But so at the ANU, I did that sort of work, cloning bacterial genes, and I was very lucky to work with some very, very smart people down there. And then my PhD supervisor, one of them, his name was John Shine, and he's a very, very famous molecular biologist. His name is on the Shine Dalgarno sequence, which is the sequence that he and Lynn Dalgarno discovered that makes ribosomes bind to bacterial DNA so that it knows where to translate proteins. Yeah. And that single discovery helped the gene cloning and bacterial expression business create new medicines like insulin, for example. So all of the insulin that people use in hospitals today is made in bacteria by the process that he was involved in inventing. So for me, that was huge.
Just a question. So it was a specific sequence that was found on the RNA of the RNA that the ribosome could recognize and then translate that into a protein?
So it's actually on the DNA, but it has a complementary sequence in the RNA. So when the RNA binds to this sequence, it knows where to start. And so this was just, in the olden days when I was a boy doing my PhD, this was just amazing work. So the whole recombinant DNA field was just taking off, and Australia was big in that area through guys like John Shine.
Right. Yeah. So after your research, your PhD, how did that transition go from biochemistry into oncology? If you could.
Yeah. So the thing that happened then was that John decided he would go to San Francisco and start a biotechnology company. And one of his great achievements is that in the space of 3 years he set up this company, and it was very successful. And he decided then he was coming back to Australia, and along with that move, he brought part of that company with him, and he set up a company in Sydney, and that's Sydney in the late 80s, and that company was called Pac Bio, and so I was really lucky to get recruited to that company, and I moved to Sydney, and we started work. And that started me working in the area of research that I'm working in now, a very long time ago. Sadly for us, the company was set up just in time for the 1987 stock market crash. And so,
great timing.
And so despite the magnificent support that we got from government, so with grants and things, the company lasted about 3 years. And we went through the process of having to dismantle the company and all of that, which is, it was a very interesting process to go through. I can't say I'd wish it on people, but as an experience, it certainly taught me a lot about the other side of that process. We hear a lot about the wonderful achievements of the biotech industry, but those achievements are built on a lot of things that start out well and don't actually do so well.
It also, I guess, highlights a whole nother side to scientific research that a lot of people don't think about, which is that fight for funding and always trying to pay your way, right? Because science costs a lot of money, but there's a whole nother side to it, isn't there?
Yeah, they're absolutely right. I had the experience of talking to one of the people who worked in a very large, very large medical companies in the United States, and he was showing me around, and he said to me, "So here's our research campus," and I said, "Oh, so how much money do you get for doing this sort of stuff?" And he quoted a number, and it turned out to be about 3 times the size of the NH and MRC budget. And I just said to him, "Well, that is amazing that you've got that kind of investment in what you're doing." And he then said, "Well, that's what we'd describe as a pimple on the back of one of the senior vice presidents of our company."
Yeah.
So that really said to me if you're talking just money and investment, the idea that Australian research punches above its weight is fundamentally true. Because what we do with the money that we have available and
It's phenomenal.
being able to be competitive is a remarkable thing.
Yeah, it makes you think what we could do with more money then as well, isn't it? Obviously.
Well, yes.
Yeah.
I've also spent a lot of time advocating for medical research through a group called the Australian Society for Medical Research, and I was very much involved with them in my younger days. And you get to meet politicians and you get to meet a whole range of people who advocate for research. And yes, without doubt, we could do a lot more with more money. The fact is that we in Australia do about 3% of the world's research, medical research.
That's actually an amazingly high figure.
Yeah, it is. And my experience with the ASMR taught me that there are an enormous number of advocates for medical research, but like everything else, it's a question of if you've got 10 very worthy things to fund and you've got enough money to fund 2 of them, which ones do you choose? So yes, we need more money, there's no doubt about that, but for us to go on and say that money needs to come from the government, and we need to say all of those things, but the whole idea of what we're doing here now at Liverpool is really about translational research, and that's about building capacity so that we can expand the overall size of investment into medical research through broadening the people that are willing to invest or are able to invest.
And that's by creating therapies or showing the utility of medical research by actually getting outcomes for patients.
Yep, that's right. Well, I'll step back just to get back how I ended up in oncology. So I told you that I kind of tend to move around a bit, but so the biotech company folded, but the basic project that I was working on was a really tiny enzyme called PLA2 at that time, and that enzyme has kept food on my table for the last 35 years. And
that's a good way of putting it.
And that to me is a remarkable thing. And so I worked with some very bright PhD students who discovered a small peptide that was able to inhibit this protein. This protein was of interest because it was induced in inflammation. It was associated with many inflammatory diseases like rheumatoid arthritis, for example. I spent a long time working on that. So it's upregulated in that, and when you do the knockout mice, which is a whole story in itself, this protein seems to be important in the inflammation that's caused by immune-mediated factors. So that then suggested that the target itself was worth working on. A PhD student at the time, Albert Singh, came to me with a small peptide that he had managed to extract from this small protein, and he said, "Look, this is an inhibitor of PLA2." And I'm thinking to myself, well, how does this work? He says it's an inhibitor because it binds to the amino terminus of PLA2, and that's where all the action is with respect to the enzyme. So it should be an inhibitor. So he then went and did experiments, and sure enough, it seemed that was the case. Now, I was pretty sceptical about it, so I made him do those experiments 11 times. Can you believe that?
Yeah.
And to his credit, he went and did it. Mind you, he said a few things about his supervisor that I won't repeat here. But nonetheless, he went and did it, and it turned out that yes, it was an inhibitor. But it wasn't really weak, so it was really, really weak inhibitor. And so we talked to structural biologists and protein chemists, and he devised ways of making that inhibitor more potent. And so we ended up, after several years working on this, with a small cyclic peptide called C2. And that inhibitor was better than the original inhibitor that he discovered. Now, we were interested in arthritis then at that time, and I was working away at that, and then I got a visit from a guy who's now a professor out of Western Sydney University called Shine Dalgarna, and he says to me, "Oh, look, I've been working on this tumour suppressor for a long time, and it's called an Exin-2. And I've just been looking through the literature, and I see that an Exin-2 modulates PLA2 action. And I saw that you work on PLA2, and you're not very far away, so I thought I'd come and see you."
So correct me if I'm wrong, but at this stage you weren't really aware or only had a cursory kind of understanding of PLA2's role in cancer or prostate cancer?
It had no role in cancer at the time. We had no evidence at all that this thing was important, apart from one publication that said that the expression of this enzyme is associated with prostate cancer tissue. So if you search the literature, you would see this one paper that says, when people have prostate cancer, you can do sections of their tumours and you'll find this enzyme there where it shouldn't be. And so that's Shihan said to me, well, if an Exin-2 binds PLA2 and it's a tumour suppressor, then why don't we try your inhibitor, because it might work as
have the same mechanism. Yeah.
And so I'm thinking, I seem to be sceptical most of the time.
That's good. So you're a good scientist then, in other words.
It's kind of you to say. I don't know. I'll leave that to others to decide. But so so I immediately started thinking about all the reasons why this couldn't be, and then in the end I thought, well, why don't we just try it? And we did, and it seemed this thing seemed to inhibit the growth of prostate cancer cells, and we then went on to look at in laboratory models of prostate tumours, and this compound seemed to make the tumours shrink, and it just kept on happening like this. And so I spend a lot of time trying to, in science, you don't want to spend time working on something that's not real for longer than you have to. And so I spend a lot of time trying to say, well, all right, so how can we test this to make, to find out that it's not really going to work for us? And so I put a lot of effort into trying to devise experiments that would really test that, and so far I haven't been able to find one.
I wanted to maybe zoom in a little bit. Let's just wait till Hamid, just gets his the charger. It's not, mine's not charging either.
A few moments later.
Okay, ladies and gentlemen, we are back. Back to the party. I was just going to ask if you could just talk about the link between inflammation and cancer, because previously you said that PLA2, which is involved in inflammation, wasn't linked to cancer itself.
So that's true. We didn't know much about that link, but as it turns out, in the last decade, the cancer researchers around the world have really established a strong link between inflammation and as a driver, and what they call an epidriver of tumorigenesis. So the classic paradigm is that cancer is caused by the accumulation of mutations, and that those mutations drive tumorigenesis, and that's certainly true for a proportion of cancers. But one of the struggles in cancer research is that you keep coming across these what they call sporadic tumours, and they're tumours that don't seem to have the mutations that everyone knows about, and so there's this whole world out there of ignorance, really, that we're trying to figure out how is it that these cancers can be around when they don't have the classic paradigm
Interesting, yeah.
mutations that we all know about. And so over the last decade, it has become very clear that the inflammatory process can act as a driver of inflammation as well. It's not
Just for people that may not understand, inflammation is a lot more than just I got a red bump in immunology, isn't it?
That's right.
Yeah, it means a whole lot of very specific things.
Well,
we probably don't have to go through all of them,
but as a biochemist, I really don't like the term inflammation because it's a clinical term.
Yeah.
Right? It's a term that was invented to describe a red bump, so it's more, it gets hot, it gets red, it gets painful, and you can also lose function. They're the four defined clinical definitions of inflammation, right? Yeah. But what people have, since the biotech revolution, people have started looking at what are the genes that are involved in this, and it turns out to be a whole range of different genes that mediate these functions. And the inflammation is a normal process as well. So when I get a zit on my cheek, well, it swells up, but it resolves. And so the biology of how that process works is really only now starting to be explored, and it's a very, very exciting time.
I was just going to ask, so maybe talk a little bit about like how inflammation actually leads to cancer. I know we want to avoid like the hardcore biochemistry of it. Are there certain compounds or molecules that are produced that then cause DNA damage? What sort of things actually happen?
Yeah, so all of the above, really.
Isn't that always the way with science?
So the thing is that, there are, as biochemists in the past, we tend to think about pathways as being, we start here and we finish here. And
so they're linear, stepwise.
Glucose falls out the end, and there's feedback loops that might control some of that. But since people like Barabasi have been doing work on scale-free networks, for example, you find, and proteomics and genomics have really started to define what's inside a cell, basically, from those points of view. You find that all of these things are linked together, and you can find that the DNA damage pathway may well be influenced externally from a pathway that is upregulated by inflammatory mediators that come from somewhere else. And so when I said all of the above, just what I really mean is that our understanding of the complexity of the biochemistry of a cell has really led us to think that there are multiple ways in which tumours can be formed, and that's really the struggle that we face in treating cancer, because we've got these great therapies that can wipe a cancer out. Sorry,
you're right.
We can wipe a cancer out completely, but they tend to have side effects because they're not specific. And we've got these great therapies that are specific and have few side effects, but they don't work on everybody. And
they only work on specific cancer types.
And even then, the cancer types they work on can become resistant to these things. And so we're really, because tumours can evolve, and so to my mind, cancer really is about the dysfunction of homeostatic regulation. So we've got a cell that it becomes dysregulated, and that can happen in several ways with mutations, for example. And that cell then isn't the kidney cell that it used to be. And so it starts to grow and do crazy things.
There's a meme out there. So Alex and I are both part of some far out Facebook groups. We just want to see how people think when they're like in these echo chambers. Like I'm part of anti-vax groups. I've come across people who think that cancer's been cured and it's been and that cure has been sequestered by pharmaceutical companies. There's an interesting meme that gets passed around where there's this girl that says cancer's been cured. You think the pharmaceutical companies will let you actually let that cure get out because they want to make money. What are your thoughts on that?
So I know some oncologists, and one thing I can tell you for sure, and I get sort of emotional about this, those folks, if there were a cure for cancer, they would make sure that people got it.
Yeah. It's absurd, really, isn't it? Because like the oncologists and the scientists and the doctors and the nurses and the whole health industry are people too, right? And cancer kills a lot of people, and these people are affected by it as well. So it's kind of makes no sense at all. But almost not even just on that side. For me, that view that taking in regard what you've just said about cancers being very individualized and personal and evolving over time, to say that like they're hiding a cure to cancer almost makes no sense scientifically, because there may not be a single cure to cancer.
There's certainly, we can say with certainty that there won't be.
So it's a false assumption right off the get-go.
And you can see that clinically today. So in the clinic, people aren't talking about single therapies for tumours. They're talking about combination therapies that target different pathways and showing that these combinations work better than either of these two compounds alone. And so that is the thinking that's guiding clinical therapy treatment right now. And the other point I'd make sitting here is that there's so much wonderful basic science that is related to this, but to the process of translating that into clinical utility is far more complicated than guys like me would ever have thought. Because there are a whole range of things that clinicians need to consider when they devise a treatment plan, and it's not about for them it's not about I'll try this inhibitor that blocks this enzyme or this it's a far more complex process than this. So they're really at the pointy end, the practical end of trying to advance the utility of all the science that we do.
Yeah, and maybe this is actually a good segue, because when we met you downstairs in the foyer here, I'd never been to the Ingham Institute. We're sitting in the Ingham Institute at Liverpool here, for people who don't know yet. And you were giving us a bit of a tour and showing us what's on every floor, and it really highlights that translational aspect that you just mentioned, and taking in almost like a holistic approach, where we take in, you said they've got places for nurses training and doing experiments to see better ways to nurse and treat patients, and also you were telling us about there's psychologists who continually using evidence-based methodologies to try and work out the best ways to communicate with patients, and then there's obviously the hardcore science part of it as well. So maybe you could speak a little bit about that.
Well, that's been one of the great excitements for me to come to Liverpool. I came here because of the opportunity that was presented to be able to translate my 20 years of bench science into a therapy that can be tested to see whether or not it's useful, and that's one thing. But the other thing is, since coming here, it's exposed me to the breadth of research that's going on in the health system, and it's not just about we're trying to invent new therapies, to cure cancer. So the idea of curing cancer is miles away from what a lot of the researchers in this building deal with every day. They're asking questions about how can I help the people that are on, say, a cancer journey better to cope with that journey. How can I, and they're using, as you say, scientific approaches, not just to come up with ideas and we'll try this or that, but to seriously evaluate the effects of those interventions. And that kind of research, is something that I just hadn't been exposed to prior to coming here.
Yeah, and it can be incredibly important because you were mentioning when we were talking, just for example, about researching better ways of nursing. You don't think about it, but if the nurses got that patient contact. So you have a treatment and you have a patient, and they're kind of divorced from each other, and the nurse is that conduit. Thank you. Conduit between the two, right?
Yeah.
And that's a really important conduit because some treatments might need to be administered at particular times, and that might not just be the morning.
Yeah.
That might be patient dependent and might change but individual patients. So better nursing can really have, I guess, a huge impact on patient outcomes. It's not just develop a drug and give it to people.
That's right. And that's another aspect of working here, that the hospital has just recently opened an early phase clinical trials unit, and so I get to talk to people who talk to patients all the time, and now, and so we're really actually starting to build those sorts of relationships that give you insight into that process. And I get to talk to people with cancer, and I was talking to one of those guys the other day who had prostate cancer, and we're just chatting about stuff, and I say, well, so you're here getting therapy, what's all that about, and what's it like? And he said, well, the worst thing about having cancer is just waiting. You get these treatments, and they're really helping, but there's so much time where you're just waiting around for something to happen, and it would just be great to have something to do while I was waiting here. And so one of the ideas in my dreams that I have about units like early phase clinical trial centres is that if we can embed the instrumentation that we use to do the science in those facilities, then patients can start to see what goes on. So they can see what goes on with my blood sample when it's taken out of my arm.
Because traditionally, they would, in a clinical trial, and correct me if I'm wrong, they would be like taking a blood sample, and then that might get sent somewhere else across the city, or even overseas.
That's right.
Where the institute that's set up here, it's a lot more direct, so it has scientific benefits, but then it also has benefits because patients can see straight away like what's happening with their samples.
Yeah, so in an ideal world, they can you can chat to people like us who do these sorts of things and find out more about what goes on.
Yeah, that's awesome.
And that would mitigate the stress of the process, because they're I think a lot of the stress and anxiety comes from the unknown and not knowing what's happening, and so being involved in that process would help them cope better in that process.
Yeah, I think so. That's my view, and I think a lot of people, as generally in the health system, are moving in that direction too. It's about patients having more control and being more involved.
Yeah.
Could we talk about like drug development? Because
well, that's what I wanted to talk about, getting that process, right? So you have this. You
Still my question.
Yeah,
Alex.
yeah. Well, go on. You set it up. You set it up. I'll defer.
I was actually watching a TED talk, and they were talking about how much pharmaceutical companies actually put into developing drugs, $15 billion over 10, 15 years of research. And they were coming up with different models. They had these almost like flow cell things that they could connect that had analogs of the brain, the liver, different organ systems. But it really got me thinking, that this is a slow process. Obviously, that's necessary because you want to make sure that it is effective and it is as safe as possible for the patients. I'm sure a lot of people don't know what that process involves, from actually finding a drug that you'd be
that inhibits
or an inhibitor or some sort of compound that you'd be interested in, and actually going through the process of developing that, or research, going through all the different phases to making that available for patients. I know I did a unit in my third year molecular medicine where they were saying, you can do many types of approaches. You can screen like 500 different compounds in these 96-well plates or whatever, and then you check which one it seems more promising, and then you do that multiple rounds until you find a compound. But then there's many other barriers that you have to overcome in order for that to actually reach the market. If you could talk about that a little bit.
So there are lots of ways you could talk about this, but I'm going to talk about it in terms of risk. And when people talk about the pharmaceutical companies, not being able or not doing the right thing, one of the things that I think about all the time is the risk involved, and that can be talked about in many ways, but let's talk about financial risk, for example, involved in developing a drug. So when I started out a very long time ago, the chances of my small little peptide getting to the front doors of a clinical trials unit, where it is now, were 1,000 to 1. And so, if I were to go to ask people to give me money back then to invest in this process, and I'd be forced to tell them, look, the chances of you getting your money back are about 1,000 to 1, and they'd be going, well, maybe I should put my money on the 100 to 1 shot in the Melbourne Cup, and I might actually have 10 times the chance of getting my money back. And so, but the process of drug development in terms of risk is about minimizing that risk over time. And so every time you do what you've just said, you develop a better compound, you do screens and you find something that works better, then that risk starts to decrease. And then there are some key elements that you need to have. First, you've got to show that the target is a genuine target, and that's done with a lot of epidemiology type work and the sort of work I was talking about before, making sections and having a rationale for intervention. Then you've got to have a compound that works. Then you've got to show that compound works in a setting that tries to mimic the clinical setting. So in my case, I need to show that it works in tumours. You then need to start thinking about, well, how do I deliver that compound? So, I could be working on a compound that works really well on cells in a dish, but when I give it to somebody, well, it doesn't get absorbed. If I decide I want to have an oral therapy, well, it doesn't get absorbed. And there are a lot of things out there already that do that sort of thing. Or if I give it intravenously, its half-life is only 35 seconds. And so there are a whole range of questions that become important. And so in my case, I just thought that it would always be good to be able to take a pill rather than having to have an injection, because I'm afraid of pain most of the time. And so we started working on, well, can this compound be orally absorbed? And so, we did experiments, and the answer was yes. It's not, that it can be orally absorbed. And so then you think, well, maybe it can be an oral therapy. And so you start asking questions about, well, if I give it as an oral therapy, will it make tumours grow less quickly? And the answer to that was yes as well. And so then the picture becomes a bit clearer as to what this medicine, this potential medicine can or can't do. And so then you've got to start thinking about issues of scale. So let's say we get this compound and it works really well and it can be orally available and it can help tumours slow down. You need to be able to make enough of it for it to be a useful medicine. And so in our case, it turns out that there are companies around the world that make this sort of stuff all the time. And I'm going, well, I've been working with 5 milligrams of this compound for a long time. I'm going to need quite a bit more if I were thinking about a clinical trial. And so you ring up these companies and they say, well, how many kilograms would you like? And, and I go, well, how much will it cost? And then they say, oh, and then I go, right, well, we won't be getting kilograms, thanks. It might be that we can afford to buy a few grams. And so that process, so the cost of production and the scalability of production then becomes a really important issue. And so then you go, in my case, we were able to get a small amount made that was going to be enough for us to do all of the preclinical analysis that you've got to do before you can even apply to an ethics committee to get an approval. And so you've got to do all that on the same batch of compounds, right? So you've got to have enough of this made so that it can be stored while you're doing all of this preclinical work to show that it's not going to be toxic, that it's going to be efficacious, that it's behaving the way it should, and all of those things on the batch of material that you hope to go into the clinic. Meanwhile, your batch material is sitting in the freezer somewhere, and then you start thinking, well, how do I know it's actually going to be the compound I think it is by the time all the stuff is done? Yeah. So stability becomes a really important question. And so you've got to start thinking about, well, is this compound stable? Is it and so we did studies with stability at minus 80 degrees, and it worked fine, and over many years. I'm not very good at this anyway.
That's okay.
Over many years, and that's great. But then you get to the point, well, how many patients have got a minus 80 freezer at home? It needs to be stable either in the fridge or on the bench. Yeah. And so you've got to go and show that all of that's possibility, or all of that can work for your compound. Then you've got to know where the compound goes. So say I give it to somebody, what happens to it? Yeah. So how is it excreted? How long does it stay around in circulation? So all of these things need to be worked out, and toxicity needs to be worked out as well on the batch of material that you've got. And so over a period of time you get that, and then you can apply to the ethics committee to have your drug put into the clinic. And then the ethics committee goes, well, you haven't shown this, and you haven't shown that, and you haven't shown, and we need you to be able to do all these things because we are ultimately responsible. Yeah. And so you then go into a process of talking with an ethics committee for, in our case, it was quite a long time. In other cases, it's not quite so long. But that all comes back to that issue of risk. So this compound that we've developed has never been given to a human person before. In fact, it was given to somebody yesterday for the very first time. And fortunately, we know that person is still with us today because, and there are good reasons for expecting that to happen. But that's the ethics committee carries all of that responsibility. And there are examples of trials where things can go wrong. Yeah. And so all of that process needs to be put in place. And then finally, sometime last year, we got ethics approval to do this.
And this
study.
Is just a phase 1 clinical trial
well,
as well, isn't it?
This isn't even phase 1. This is what we call phase 0. Yeah. And the reason it's phase 0 is because a phase 1 trial usually looks for a maximum tolerated dose. And we didn't have enough compound to do that. So what we're doing is a limited dose escalation study, and from that we'll be able to get a lot of information that will make a proper, phase 1 study that will inform that process.
Just because we're kind of coming to the end of this, and I know you probably have to go and have meetings and have actual work, unlike Alex and I.
You guys do important jobs, you should be very happy with what you're doing.
Just briefly, what is the difference between phase 1, phase 2, phase 3? If you could just
okay, so phase 1 is all about safety, and there are two things you want to do in a phase 1 trial. You want to show that it's safe. You want to also get pharmacokinetic information about the way the drug behaves, and you
dosage and things.
Yeah, because that then informs the dosing for future trials. Phase 2 trials are done on more patients, and you're looking at specifically at efficacy then. Does this compound work? And then if you get positive results from that, you do a phase 3 study, which is a much bigger multi-site trial on a much larger number of patients, again looking for efficacy endpoints. And then in phase 4, once something's marketed, there's still monitoring that goes on. So the drug gets marketed. There are phase 4 studies that look at its long-term safety and monitor its progress over time. And that then again is about reducing risk. So even if this compound that I'm working on is successful in this phase 0 trial, the risk will be reduced to about, say 10 to 20 to 1. So now I'm in the ballpark of saying to people, well, if you give me your money, it's a bit like putting your money
money on
a horse. And in every step of the way in the clinical trials process, you reduce the risk. But the thing about it is it's still very risky. So you can get to phase 3 and you've still only got a 50-50 chance of being successful. So, it's a toss of a coin. So when people say that pharmaceutical companies are messing us around, it's easy to make that argument, and in some cases they might be right. But overall, the process is really complicated.
Tedious, yeah. Do you have any other questions before we move on to the last two?
I was just thinking, assuming all this is successful, because I was reading somewhere the Ingham Institute has this wall of names, so maybe you can tell us about that. But assuming a best case scenario for C2, what are the likely projections of how many people? Because I think I was reading somewhere that almost 3,000 Australian men die every year from prostate cancer. So how effective are you expecting C2 to be if all goes really well?
Look, all going well, in advanced prostate cancer, from our animal data, we would expect that this compound, if everything goes swimmingly well, would save about 1,000 Australian lives a year.
See, that to me is really amazing because you think about people who are paramedics and doctors and things who are saving lives every single day. But in a sense, that almost pales in comparison to a researcher who can get through all of this incredible hard work that you've just described to us, and then have a drug or a treatment come out of that and literally be responsible for saving 1,000 people every single year.
Yeah, look, it's hard to compare those two things. I'd never mess with paramedics.
Don't get me wrong. Yeah, don't get me wrong. I think it's hard because doctors and paramedics it's a very direct. Where for researchers, particularly in medical science, it's quite indirect. But it's still there nonetheless.
Yeah, that's right. And I get back to the risk argument again. Paramedics and doctors can't afford to take the risk. They don't. They don't have that luxury. People like me get the luxury of being able to try things that may not work, and that's one of the great opportunities that you have in a scientific career, and it's something that's undervalued a lot. But for me, it's just gold, because you get to test ideas, and it's okay if they don't work. But if you're a paramedic or a doctor, you don't have that luxury. And so I just think it's just I get it's just a great lucky thing that this whole process has happened to me. I'm a kind of in the Tim Minchin's idea that that lots of things are just lucky. The fact that we're here is lucky. The fact that we can understand what we understand is a lucky thing. None of this has to happen. And the idea in a scientific career, the other idea that he talks about is being micro-ambitious. You know, I didn't start out this process wanting to cure prostate cancer. You know, all I wanted to do was pass my exams and get my PhD, and you know, and so these discoveries get built on tiny advances that if you focus on them and work really hard at what you're doing, you just don't know what can happen. And so that, I think, is one of the great benefits of being in the science business, for sure.
That's awesome.
Last two questions, and we'll end it with this. Looking into the future, what are you most afraid of? And this can be in any context. And the flip side of that, what are you most hopeful about?
So, in terms of, I mean, right now, you know, what I'm most afraid of overall is, that's an interesting question. So with respect to my science, I don't want to hurt anyone. That's a concern. And so, in my dreams, if we can get through this trial without hurting anyone, that would be just fantastic. There are lots of other broader issues that we could talk about in that way, but with respect to this work, that's the number one thing. In terms of what I'm most hopeful about, I am enormously hopeful about the future of our young people. And you know, when I went to school and went to university, education was free. And I look at my daughters and younger folks today, and they've got to pay for their education, and there's all these issues for them that I certainly didn't have to face at all. But they are unbelievably smart. And I have enormous hope for the future because I know some of these people.
That's awesome.
It's a beautiful answer. Thank you so much for your time. We really appreciate talking to you.
It's a fascinating conversation.
And very informative.
Thanks a
Well,
lot.
thanks for asking me, because I'm learning to talk more.
Great.